Hello, welcome to my project. It's titled Identifying and Characterizing Gene Co-Expression Modules Underlying Resistance to Androgen Deprivation Therapy in Prostate Cancer. The transition from unicellular to multicellular life was a key evolutionary event requiring cells to coordinate and communicate with each other to ensure an organism's survival. This new cooperation was achieved by acquiring new genes, forming gene regulatory networks to coordinate and regulate cellular processes. This coordination is essential in maintaining an organism's survival, and the dysregulation of these cellular processes results in cancer. Previous research has shown that regulatory networks are often dysregulated in cancer, allowing tumors to survive and progress. This dysregulation subsequently results in many of the hallmarks of cancer, which are a key set of features common across all cancer, which become more pronounced as the disease progresses. However, another way we can view these hallmarks of cancer is as tumors losing features of multicellularity, expressing more unicellular features as the disease progresses. This is best explained by the atavism hypothesis of cancer, which describes cancer as a failure of fundamental cellular processes, reactivating transcriptional programs evolved in more ancient unicellular organisms. A notable example of multicellularity loss is found in prostate cancer, where primary tumors are graded using a histological Gleason grade measuring tumor dedifferentiation, which is correlated with the worst prognosis. Previous research within our group revealed as prostate tumors increase Gleason grades, they preferentially express more unicellular genes, likely due to a coordinated breakdown in gene regulatory networks required to sustain multicellularity. This coordinated breakdown may have prognostic value, enabling us to understand the underlying mechanisms driving prostate cancer progression. By building on this, we aim to characterize how gene regulatory networks change from normal prostate tissue to form tumors, and furthermore, how these gene regulatory networks change at multiple stages of prostate cancer, enabling us to identify gene expression signatures of tumor behavior, which has immense clinical value. Using a computational approach, we will develop an evolutionary network analysis pipeline to characterize changes in gene regulatory networks across multiple stages of progression to build a comprehensive landscape of prostate cancer. First, using gene expression data from prostate cancer patient samples, I will identify co-expressed genes using a weighted correlation network analysis, which calculates gene expression across multiple samples and then groups highly correlated genes into co-expression modules. We then apply gene ages to genes within co-expression modules using phylostratigraphy to derive gene ages by tracing the evolutionary origin of orthologs to major evolutionary events separated into unicellular organisms such as bacteria and yeasts, and multicellular organisms such as animals and humans. We can then characterize network topologies of genes within co-expression modules to understand how genes regulate each other, and then perform functional enrichment to understand how these activated pathways relate to co-expressed genes. Preliminary results using primary prostate cancer patient samples from TCGA, stratified by Gleason group, has shown the capability of our pipeline to identify changes in gene regulatory networks across samples. By first calculating gene age enrichment using the proportion of gene ages within co-expression modules, we identified modules as unicellular, multicellular, or mixed, where there was both unicellular and multicellular genes. These mixed modules are the most interesting, as this is where we would expect to see most of the new connections being formed between unicellular and multicellular genes. Comparing gene age enrichment in primary prostate tumors from Gleason group 1 to Gleason group 5, we were able to identify distinct co-expression changes indicating gene regulatory networks increasingly breaking down and rewiring as prostate tumors progress to higher Gleason groups. More specifically, first, we see gene regulatory networks begin to break down, becoming more fragmented as tumors progress, resulting in more co-expression modules. And second, new connections being formed as a result of gene regulatory networks rewiring, indicated by an increased number of mixed modules. These early results give us confidence in our pipeline's ability to characterize changes in gene regulatory networks across multiple stages of prostate cancer. Our pipeline is still in development and is constantly improving. We are currently working on implementing network topology analysis to understand how genes regulate each other, focusing on the connections between unicellular and multicellular genes. We will then work on implementing functional enrichment analysis to identify how these activated pathways relate to the co-expressed genes. This study presents a new paradigm in prostate cancer biology, which aims to understand how gene regulatory networks maintaining multicellularity are broken down in prostate cancer, driving tumor progression by activating unicellular pathways. If you would like to know more, please have a look at my poster, send me a message, or email me. Otherwise, thank you for watching.